So in the last video, I thought that it might have been yeast that I saw under the microscope, because I used brewer's yeast and I put it in the toaster to kill it and turn it into yeast peptone as one of the ingredients in my culture, and I worried that it might not have killed all the yeast. So what I'm going to do is I'm going to take a little bit of yeast, put it on a slide, add a little bit of water, and check out yeast under the microscope to see if it fits the bill or description. A little bit of yeast, a little bit of sugar so that they have some energy to do yeasty stuff, and a little bit of water, not too much. I've got this damp ball, and just mix it together with a relatively sterile thing. This isn't super precise science. However, yeah, I'm gonna mix. And now I'm gonna add the little cover slip onto it. Unfortunately, it's plastic since it's the only one I have right now, but it should work. And I'm gonna dab off the excess. Dab off the excess with some paper. It is at 64x magnification. As you can see, some of the crystals of sugar and yeast haven't quite dissolved yet. However, I think it's gonna be good enough. I'm gonna crank up the magnification to 160x. So this is 640x without a dark field, but with the polarization on. Now I'm gonna add the dark field and align it. Disaster experiments here. So, I have been on coronavirus quarantine. My town has been, so I haven't had a lot to do. I have to do school online. But it's not as much as I would have had to do since they are going to shorten all of the Regents exams and stuff. So, to keep busy, I have been making more YouTube videos than usual. And, stay tuned because I'm going to probably continue making videos at a faster rate than usual until this thing blows over. I would talk about the coronavirus, but I tried to make a video on that, and I don't have a lot to say that hasn't already been said. Yeah, hope you enjoyed, and see you in the next video. You have 2 samples of bacteria. In my collection, I have root bacteria, and I also have cholera. So I'm going to check out both of those. Cholera at 640x. Little dots. So yeah, it does look a lot like the sample of bacteria that I had. They're both sense, considering that both cholera and Y. enterocollitica / Aeronomus hydrophila, I'm not sure which species it is. Both species are gram-negative rod-shaped. However, cholera is known to have like spiral shape as well. A little bit. Like the cholera, this root bacteria is stained, and you can see, I think they used fuchsia red, like the red that you use to stain gram-negative bacteria. So anyway, yeah, I think that the samples that I got from the petri dish were a lot closer to the bacteria than to the yeast. That one specific time that I showed you, yeah. So I hope you enjoyed, and see you in the next video.